Hi, my name is Andre, and I'm the PI of the Laboratory of Biosystem Dynamics. I'm here today to present you our work into understanding how do bacteria activate transcriptional programs that allows them to become more adapted to cold shock environments. We focus on understanding how gene regulatory mechanisms control their response to specific stresses. Nowadays, we combine various techniques and knowledge. For example, we use strain libraries with fluorescent proteins to verify if the genes are doing what we expect from them or to study what they do for the first time. For that, we use machines like flow cytometers and microscopes. Then we try to understand how can we explain their response. For example, maybe they have something special in their sequence. We also try to understand what are they doing? What are specific genes doing? So we try to study the ontology of responsive genes to stresses. Overall, our goals are to produce new dynamic models of the gene regulatory mechanisms and then try to apply those mechanisms in new synthetic genes or use those mechanisms, for example, to target bacteria with new antibiotics. The main reason to study why bacteria or what The main reason why we study cold shock is because this is a critical perturbation that gut bacteria have to survive if they're going to remain in the biosphere. For example, a gut bacteria, the standard cycle that it goes through is it starts, for example, in the stomach of a mammal like a cow, and then the cow at some point defecates and some of bacteria pass to the soil where it can get very, very cold. So this must be survived if it's to enter, for example, a plant and then be once again ingested by the cow, thus returning to the stomach. And what we want to understand is how has the gene network of E. coli evolved in order to increase as much as possible their chances to survival to cold shocks. In simple terms, this is what we expect. We expect that bacteria has evolved its genes regulatory mechanisms in order to produce some sort of transcriptional program over time once subject to cold shock that will activate and repress certain genes which will change what is happening inside the cell and increase its chances of survival. It is these mechanisms that we want to study. And from past studies, we knew that there's a phenomenon that occurs in DNA that may be associated with the transcriptional response program to cold shock. The DNA is kind of like a rope, and it's usually in a negative coiled state. So in order for the DNA to be read, it has to be opened for reading. And to do that, you have to pressure that rope and increase the coils in the back and the front of the RNA polymerase. And to compensate for this, the bacteria has evolved certain proteins called topoisomerases, one of them named the DNA gyrase. And the DNA gyrase removes positive coils. Meanwhile, another topoisomerase removes negative coils. But there's an interesting imbalance in this system, which is to remove negative coils, no energy is needed, but to remove positive coils, energy is needed. And that energy is not for free. The cell has to do a lot to produce that energy, and it might not be very available in situations where the environment is not favorable. And it was this that we hypothesized. We hypothesized that something happens in supercoiling regulation, that will make this process harder. And so if a gene is more subject to being locked due to positive supercoiling buildup, because if it gets too coiled, it won't be easily open for transcription. So such genes which are more subject to positive supercoiling, maybe they get fully repressed during cold shock. And we went to study based on that hypothesis. We hypothesized that many cold shock repressed genes could become locked during cold shock due to changes in DNA supercoiling, since gyrates needs ATP to remove positive supercoiling buildups. We thus performed the following. We started by finding out which genes were strongly repressed during cold shock. For that, we perform an RNA-seq, which allows you to determine if a gene significantly changed their activity in a positive or negative way compared to a control condition. Once having identified which genes change significantly in total RNA numbers inside the cell, we went and performed flow cytometry, which allows us to see what happens to the protein numbers of those genes. We use a YFP strain library and flow cytometry because that provides you a lot of sensitivity to the number of proteins, and you can see it in many cells. So you see changes in both mean levels as well as in cell-to-cell variability. Having classified genes as supercoiling sensitive, we were then able to perform a different test where we instead subject cells to the antibiotic novobiocin, and that blocks DNA gyrase from curating the positive supercoiling. And we ask ourselves, so which genes were subject, changed more their activity, again by RNA-seq, when the cell was subject to novobiocin? Because it ought to be also the positive supercoiling sensitive genes. And then we compared the results when subject to cold shock and when subject to novobiocin. And genes part of both cohorts most likely are genes which are cold shock responsive, of course, but that they are repressed because of their supercoiling sensitivity. And then, of course, we performed other tests. Particularly, we went and performed measurements of biophysical parameters, such as if in fact positive supercoiling has accumulated in cold shock, then we should to some extent see that in the DNA conformation, in the nucleoid structure or biophysical properties during cold. We also went, of course, verify if in fact there's, for example, a lack in ATP energy inside the cell during cold shock. Finally, having verified our cold shock response mechanism, we produced a stochastic model of that process, which can be used in a multitude of situations that we will describe later. But of course, in addition to our hypothesis, which is that ATP is lacking, so gyrase is having problems operating, which will affect positive supercoiling sensitive genes, there's a lot of other possibilities of what could be happening in the cell at that time. For one, maybe other topoisomerases are being affected, or their numbers. Maybe instead it's the RNA polymerase that is being affected by the cold shock, like it's lacking or it exists in much larger numbers. And so we started by measuring the levels of topoisomerases and RNA polymerase, which are shown here in this graph of their relative values compared to the control condition using flow cytometry. And clearly from these results over time after the cold shock occurring, we see that after 80 minutes there's still very few changes in the total numbers. And so it's not these numbers that are causing anything, most likely is in the efficiency with which they are operating. Something that also needs to be considered because it affects many genes and we are looking for global responses to cold shock are transcription factors. So we asked ourselves, are input and output genes of transcription factors correlated in their response over time to cold shock? So once applying cold shock, we waited 20 minutes to see what happens to the genes, and then we waited another 80 minutes to see what happens to the genes. And we asked ourselves, is there a correlation between the responses of genes that produce transcription factors at 20 minutes and the response of genes that are outputs to those transcription factors at 80 minutes? And if you look at the graphs under optimal conditions and under cold shock, you see that while under optimal conditions, there clearly is a correlation in their dynamics over time. Under cold shock, there is no correlation, in fact. So in general, it seems that transcription factors become rather ineffective under cold shock conditions, which was quite surprising. So we then asked ourselves, what is happening concerning protein numbers in individual cells? Because if there's any change in the mechanisms of protein production or protein degradation, most likely there will be a change not only in mean levels of proteins, but also in the cell-to-cell variability in protein levels. And we compared protein by protein for several proteins that you can see. Each dot in the graph is a protein. We compared how does the mean relate to the coefficient of variation in protein numbers in individual cells for all these proteins, both in optimal temperature as well as under cold shock, 120 minutes after cold shock and 180 minutes after the cold shock. And we verified there was no change between 120 and 180 minutes, so we compiled that into the same and compared to the optimal temperature. And then we fitted a line, and you can see that the lines don't actually match for cold shock and optimal temperature data. And it is more noisy for cold shock as measured by omega, which is the product between squared coefficient of variation and the mean. Then we selected 6 proteins and we went and said and asked ourselves, so how did this change in cell-to-cell variability occurred over time? Was it instantaneous? How did this happen? And we plotted it over the 140 minutes, starting from when applying the cold shock. In the control, once again, we only used temperatures of 30, 25, and 20 degrees, while for cold shock, we used 15 and lower degrees. And you can see there that it starts the same, of course, because protein numbers take time to change. But when we reach 40 and 60 minutes, the cold shock conditions clearly cause a shift in noise in production or degradation, which we found later to be production, in fact. But interestingly, it becomes more noisy. And this tells us something about what happens at the transcription level that could cause such a drastic change of almost like 50% in omega, but only 60 minutes after the application of the cold shock. And so having this observation that the cell-to-cell variability increased, we hypothesized that, as mentioned before, that cold shock may be locking some promoters activity because it disturbs DNA supercoiling. And we consider this from the point of view of models. So we consider 3 models. The first model is a so-called one rate limiting step model. This is the simplest possible model of transcription where there's a promoter which has a certain rate of RNA production. Later on, the experiments showed in the early 80s that there was actually 2 rate limiting steps in transcription. One is a closed complex formation where the RNA polymerase has to find and bind to the promoter. But then it has to open it, which is the second step, which is relatively lengthy in time. And this makes the process a 2-step process. A third model, which was considered in other conditions, it's a so-called on-off model. There, the promoter is not always available for transcription. It can actually become locked. It jumps between on and off states, and only when it's on does RNA production occur similarly to what it would occur in the one rate limiting step model. Now, what's interesting about this model in this case is that For the same mean RNA production, which then is also affected by translation and RNA and protein degradation, for the same mean RNA production, the first model has a certain amount of noise, but then the second model, the two rate limiting step, has less noise, that is, less variability in protein numbers. And the third model, it actually has more variability in protein numbers. So for the same mean production, they differ in noise, or in other words, in variability between individual cells. And so clearly the model was identifying that there should exist during cold shock some sort of on-off mechanism in transcription in some genes that would increase the cell-to-cell variability in protein numbers. Past studies have identified that locking due to positive supercoiling could cause that. And so we subject cells to the antibiotic novobiocin, which is known to inhibit gyrase. And again, we performed RNA-seq. And then we confronted the responses to novobiocin to the responses to cold shock 20 minutes after the cold shock, because this confrontation was done at the RNA level, and that suffices for the RNA numbers to change. And you can see in graph C that when considering cold shock responsive genes, there is a very strong correlation between their responses to cold shock and their responses to novobiocin. There's even a correlation between when you consider all genes at the same time, which is very interestingly. But if you consider only genes that were highly expressed but were not cold shock responsive, you don't see that correlation. So all results pointed out that indeed there must be a similar mechanism shared by cold shock responsive genes and genes that are highly responsive to novobiocin. And then we also observed what was happening over time. So again, we used fluorescent proteins and we measured the omega, so the CV squared times the mean, over time once we subject cells to gyrase inhibition, but at standard temperatures. And again, we observed that there's this jump of 50% in the omega and at similar time scales. So at around 60, 80 minutes, they become much more what we believe to be noisier and thereby diverse between individual cells in protein numbers. We then turn our attention to positive biophysical phenomena that could further sustain our hypothesis that cold shock response genes are likely to be highly sensitive to positive supercoiling buildup, which to occur at the global level might actually affect nucleoids. And so what we did was we measured nucleoids by DAPI staining, and we evaluated those nucleoids areas relative to the cell size, and we observed that it decreased. And for that to occur, one way for that to occur is that the nucleoid is becoming more compacted, which would occur if there's more and more accumulation of positive supercoiling buildup. Interestingly, this occurred very fast, in around 15 minutes, which is consistent with when did noise in gene expression started to increase. Another biophysical parameter that we analyzed was the so-called clouds formed by gyrase inside the cells. So gyrases must be permanently binding and unbinding to the DNA in order to solve positive supercoils. We expect that if there's many coils, they're going to spend a long time bound to the DNA solving them. If there's very few, they will mostly just be binding and binding, and so there will probably be a bigger cloud, less overlapping with the DNA itself. But this is of course not easy to predict absolute values. So we try to compare with the clouds of a molecule that has a similar function, which is the RNA polymerase. It's always binding and binding, and sometimes it stays there, but in this case is to do transcription rather than to solving positive coils. And we observed something very, very interesting. So in the case of the gyrases, there is a much better overlap between the gyrase and the DNA during cold shock. So this ratio of intensities increased. Meanwhile, for the RNA polymerase measured by the beta subunit, for the RNA polymerase, that didn't happen at all. So somehow, I think that this is indicative that the way that the RNA polymerase interacts with the DNA did not change very much. Meanwhile, the way that the gyrase interacts with the DNA changed significantly. Again, supporting the idea that supercoiling is being significantly disturbed during cold shock. So why would a cell be accumulating positive supercoils but not negative? The main difference, as we said in the beginning between the two processes, one of the main differences at least, is that solving positive supercoiling is ATP dependent. It consumes ATP. So it is tangible that there's something not normal in ATP levels when compared to optimal conditions. And so we tested that. We measured cellular ATP levels during cold shock, and we observed that there's a significant decrease in these levels over time. And at 60 minutes, it's already very, very significant, and then it remains like that, more stable. So it appears that the cell reaches a new quasi-equilibrium, but to do so, it needs to have a much lower ATP level than in normal conditions. And this would explain the inability of gyrase to solve positive supercoilings, while the other molecules, the other topoisomerases, can solve the negative supercoils still. And with this, we reach the end of our presentation. We think that the main conclusion is that locking due to positive supercoiling buildup is a trigger of at least 50% of all cold shock repressed genes. And this is explainable by the very significant decrease in ATP levels during cold shocks. I believe that this study is interesting because it gives a mechanism for how genes are responding. I think nowadays the techniques allow us to relatively easily determine which genes are changing with a certain event, but it's still very hard to determine why certain genes change and others don't, for example. That is, it's hard for us to dissect the mechanism. And in this case, it appears to be clear that supercoiling asymmetries in the DNA, so differences in supercoiling sensitivities, is playing a very significant role. Maybe supercoiling is playing roles in other events as well, but certainly is interesting in this case what it's doing. From a more global perspective, E. coli is still one of the elements in the critical group of who, in what concerns the resistance to antibiotics. And so understanding the mechanisms that it uses to respond to stresses may be very assisting for us to then develop new antibiotics, because we could target those mechanisms and thereby reduce their resistance, or making that resistance very, very, very unlikely. And so I believe that continuing to study the response to stresses other than antibiotics, for example, may still give clues to new antibiotics. Moreover, the way bacteria regulate their temperature is actually something very important that influences us. For example, bacteria decides to turn on and turn off specific transcriptional programs involving many genes as a function of temperature, and thereby we need to live with it in what concerns bioreactors. If we could alter when bacteria trigger their responses to a certain temperature shifts, we may be able to actually make use of more energy efficient transcriptional programs rather than those that bacteria has evolved to favor their survival and reproducibility. which is not necessarily the most important thing when in a bioreactor. And I think that the tuning of how the bacteria itself regulates DNA supercoiling might be a very interesting road of research for this goal. Finally, I think even at at the smaller level, that is at the level of synthetic circuits, maybe we can use these features in order to control their functionality in a given condition, but not in the other condition. For example, I'm quite convinced that if we choose where to in the DNA, where to in bacterial chromosome locate the synthetic circuit, will make that circuit subject to different levels of positive supercoiling and to different changes to different perturbations. So we may be able to play with positive supercoiling buildup as a means to regulate the functionality of our synthetic circuits. And with this, I would like to end my presentation and thank you all for listening. And please contact us if you have questions or similar interests in research. Thank you so much.